imagine a future where your computer isn't built, but it's grown.
Right.
Like, I want you to step away from the idea of motherboards and silicon for a second and really visualize this. You don't just upgrade its RAM, you feed it a nutrient broth.
Yeah, you literally have to keep it alive.
Exactly. You don't just reboot it, you let it rest because it actually gets fatigued.
Yeah.
It's a machine that learns, that ages, and that undergoes what scientists call representational drift.
Which basically means it changes its mind over time.
It literally changes its mind, and eventually, like all living things, it dies. So when the hardware on your desk is actually alive, where does the machine end and, the entity begin?
Well, welcome to another My Mind MashUps deep dive, where we unravel mysteries, spark curiosity, and make sense of complex ideas together.
We want to briefly thank our coffee supporters for helping fund continued research.
Yes, and thank you for sharing your time with us today. If you enjoy content that makes you think, follow or subscribe wherever you're listening.
You can also like, share, or leave a comment to help more curious minds discover the conversation. If you would like to support the channel directly, the link is in the description.
So we are looking at a stack of incredibly recent paradigm-shifting research today.
Yeah, we've got the landmark 2022 DishBrain study, the 2023 Brainoware paper, an absolute breakthrough February 2026 Cell Reports study, and a deep dive into what the tech industry is calling wetware.
Right, and our mission today is to cut through the sensationalist headlines. We want to separate the science fiction.
I like this whole idea of fully conscious, tortured souls trapped in a digital matrix.
Exactly. We want to separate that from the very real, verified scientific breakthroughs in synthetic biological intelligence, or SBI.
Which is a crucial distinction.
It is. We are dealing with organoid intelligence here. And to even begin this conversation, we have to ground ourselves in the physical reality of the laboratory.
I'm really glad you brought that up, because my immediate mental image, and I bet I'm not alone here, is like a full-sized human brain floating in a jar of glowing liquid.
With USB cables jammed in.
Yeah, exactly. With cables coming out of the cerebral cortex. I know that's not what we're talking about, but how does a biological cell actually plug in to a digital world?
Well, let's strip away the sci-fi movie props. We are talking about an architecture called a closed-loop embodiment.
Okay.
The foundational technology is a high-density multi-electrode array, or an MEA. Picture a tiny, flat silicon grid about the size of a postage stamp. And it's absolutely packed with thousands of microscopic electronic sensors and stimulators. Researchers take living neural cells and culture them, grow them, directly on top of this grid.
Okay, stop right there. The cells are just sitting on the wires.
Essentially, yes.
How do the wires actually talk to the cells without destroying them?
It's all about translating environments into electrical language. The cells and the circuitry are practically woven together at a microscopic level.
Wow.
Conventional software takes a digital environment, say a simulated room, and translates the events in that room into tiny, precise electrical pulses. The MEA fires those pulses into the living tissue.
And the neural network physically reacts to that electrical stimulation.
Because neurons naturally communicate via electrical spikes anyway.
Right, we're just speaking their native language.
Precisely. Their biological response is picked up by the recording electrodes underneath them, decoded by the software, and translated back into a digital action in the virtual world.
Okay, I see.
And then the consequence of that action dictates the next electrical signal sent into the cells. That is the closed loop. Stimulus, biological reaction, digital translation, new stimulus.
And it happens in milliseconds, over and over. Which brings us to the 2022 DishBrain experiment led by Brett Kagan.
Yes, that was a huge milestone.
Because this is where that closed loop gets applied to something we all recognize. They took about 800,000 living human and mouse cortical neurons, grew them on one of these arrays, and plugged them into a simplified game of Pong.
They did.
But I'm trying to visualize the interface. I mean, they don't have eyes. They can't see the little digital ball bouncing across the screen.
They don't need eyes if the spatial data is encoded into the electrical pulses. The software communicated the location of the Pong ball through frequency and location.
Oh, like the faster the pulse, the closer the ball.
Exactly. And which specific electrode fired told the neurons where the ball was spatially on the grid. The output of the cells then controlled the movement of the virtual paddle.
But the training method is what really caught my attention. It borders on like the behavioral psychology we see in living animals.
It does sound like it.
Yeah, because if the cells successfully intercepted the ball, the researchers fed them a predictable, clean electrical signal. But if they missed the ball, they blasted the cells with a burst of chaotic, unpredictable static.
Right.
And over a matter of minutes, the biological network adapted. The rally links improved. The original paper actually used the word sentience in its title,
They did.
arguing that the network was actively trying to minimize that unpredictable chaos, like it felt pain or at least discomfort, and tried to avoid it.
Look, we have to be very careful with the terminology here. The use of the word sentience generated massive controversy and is absolutely not the consensus of the scientific community.
Really? But it changed its behavior to avoid the static.
Yes, but you're attributing a human emotional state pain or discomfort to a flat layer of cells. The authors were actually leaning on a theoretical framework in neuroscience called active inference.
Okay, explain active inference because it sounds like a machine learning term, not biology.
It bridges both, honestly. The theory posits that biological systems naturally act to minimize unpredictable sensory surprises. They want homeostasis.
They just want predictable input.
Exactly. The operational fact of the DishBrain study is that the network changed its behavior to improve task performance. It demonstrated biological plasticity.
But it's not suffering.
No, it is a hybrid control system. It is not a conscious gamer having a subjective emotional experience of winning or losing. It's a biological mechanism physically adapting its own connections to smooth out an engineered feedback loop.
Okay, I can accept that it's not sitting there thinking, I really need to win this match. But Pong is fundamentally a flat 2D reaction game. If we want these biological networks to process truly complex information, a flat layer of cells in a petri dish isn't going to cut it. Real brains are three-dimensional.
Which leads perfectly to the wall researchers hit and how they broke through it in February 2026.
The Cell Reports study.
Yes, this is where the field shifts from flat cultures to 3D structures and introduces genuine goal-directed learning. But I must be explicitly clear here to avoid feeding the science fiction panic.
Go ahead.
This 2026 study used mouse-derived cortical organoids. These were not human mini-brains. They were 3D spherical clusters of neural tissue, roughly the size of a pinhead, generated from mouse stem cells.
Mouse cells are not, though. The architecture is what matters. They hooked these 3D organoids up to a classic AI benchmark task.
Balancing a simulated pole on a moving cart.
Right. And instead of just punishing them with static, they used targeted adaptive training signals. If the pole started falling and the organoid made a move that didn't help stabilize it, the system delivered targeted electrical stimulation to disrupt that specific pathway.
Forcing a different neural route.
Yes. Using the structured feedback, the organoid's success rate at balancing the pole jumped from about 4.5% to 46%.
And the most important part of that study wasn't just the success rate. It was that the researchers proved exactly how the tissue was learning.
How so?
They introduced pharmacological agents that blocked glutamatergic transmission in the organoid.
Okay, hold on. Let me stop you right there. Do not throw glutamatergic transmission at me without a lifeline. What does that actually mean?
Fair enough, fair enough. Think of AMPA and NMDA receptors.
Still sounds like jargon.
Okay, think of them as the physical, biological locks on the doors between neurons. In your own brain, when you learn something new, these receptors chemically open up, allowing new connections to strengthen.
So it's the literal mechanism of memory formation.
Exactly. When the researchers chemically blocked those specific receptors in the mouse organoid, the learning completely stopped. The success rate flatlined.
That is incredible. It proves unequivocally that this wasn't some electronic artifact or a software glitch.
No, the tissue was physically, biologically remodeling its own synapses inside the 3D structure based on the video game feedback.
But I found a detail in that study that fascinated me even more. After a 45-minute rest period where the organoid wasn't stimulated, its performance dropped right back to the baseline.
The memory faded.
Yes, it just faded.
It is transient, feedback-dependent plasticity.
But think about the implication of that. It's fleeting, sure, but it proves that a living 3D biological network can execute goal-directed improvement, hold a rule, use it, and then simply let it go when it's no longer needed.
It's not a hard drive that stores a file forever until it's overwritten.
Right, it's an organic resting state.
It is a dynamic state, yes.
So we've moved from mouse organoids balancing virtual poles to processing complex human data. How do we make the leap from a balancing act to a biological computer recognizing actual human speech?
That brings us to the 2023 Brainoware system. And this is where the field shifts into a computational architecture called reservoir computing.
And in this study, they didn't use mouse cells.
No, they used a 3D human brain organoid derived from stem cells.
Right. They wanted to see if human tissue could process complex temporal data that changes over time, like audio.
Yeah.
They fed this organoid 240 distinct audio samples of Japanese vowel sounds spoken by different male and female voices. And after training, the system's ability to correctly classify who was speaking rose from about 51% to 78%.
It's a huge jump.
It is, but I am struggling with mechanics here. How is a lump of human cells recognizing the nuances of a Japanese vowel? Does it learn the language?
No, it doesn't learn language. Understanding this requires a complete shift in how we think about computing. Think of the human brain organoid, not as a tiny human sitting at a desk typing out translations, but as a complex, dynamic pond of water.
A pond. Walk me through this.
When you throw a rock into a pond, the rock being the electrical translation of the audio input, the vowel sound, it creates a wildly complex, unique pattern of ripples that bounce off each other and the edges of the pond.
Okay, I'm picturing it.
A conventional digital algorithm sits on the shore of that pond. The algorithm doesn't try to understand the water. It just learns to read those specific complex ripple patterns and associate them with a specific output, like a particular speaker's voice.
So the organoid is the reservoir.
Yes. It acts as a dynamic biological filter that transforms a simple one-dimensional audio input into an incredibly rich, high-dimensional physical state. The digital layer on the outside does the actual decoding.
I am trying to visualize the memory aspect of this pond, though. If the organoid is the water, where is the memory stored? Is it the ripples bouncing off the edges?
That is exactly what researchers call fading memory. The physical tissue of the organoid retains the electrical shape of the previous input just long enough to interact with the next input.
So if I say the word hello, by the time the O hits the organoid, the tissue is still physically vibrating, biologically speaking, from the H, E, and L.
Precise.
It naturally processes sequences of sounds because the physical state of the tissue remembers the last millisecond of input. It perfectly mimics short-term sequence processing without a computer engineer having to write a single line of code to explicitly program it.
It's just physics and biology doing what they naturally do. It exploits the natural nonlinear dynamics of living tissue. That is the elegance of reservoir computing.
If this conversation is making you think, follow or subscribe so you don't miss the next My Mind MashUps deep dive, because we really have to talk about the commercial reality of this.
You too.
If this wetware is so elegant and powerful, can a tech company just go out and buy a biological server farm right now?
The short answer is yes.
Yeah.
The infrastructure is already being commercialized.
We have companies like Cortical Labs selling the CL1 hardware. This is a system with an integrated life support environment designed to keep 2D neural cultures viable for up to 6 months.
They even launched a Cortical Cloud, offering Wetware as a service.
And then you have FinalSpark's Neuro platform. They've maintained 3D neurospheres for over 100 days. But here is the mechanical question that blows my mind.
What's that?
FinalSpark allows researchers around the world to log in remotely via Python and run experiments. How do you log into a living brain from a dorm room in another country? What actually happens to the tissue when I hit enter on my keyboard?
It is a marvel of integration. When you run a Python script on your laptop, it sends a digital command to FinalSpark servers in Switzerland. Their application programming interface, or API, translates your code into specific instructions for the microfluidic pumps to deliver certain neurochemicals, or commands the MEA hardware to fire specific electrical voltages into the exact location of the living human tissue you've rented. The tissue reacts, the sensors read the biological voltage, convert it back to digital data, and send it over the internet to your Python terminal. You are directly, physically manipulating a living neural network from thousands of miles away.
Literally Wetware as a service. But let's do a reality check, because the big marketing pitch I see everywhere is energy efficiency.
Right.
I hear over and over, the human brain runs on 20 watts of power, but an AI data center uses megawatts. Biological computers will solve the AI energy crisis. I have to assume that's a massive oversimplification.
It is deeply misleading unless you understand what is actually being measured.
Yeah.
The claim is based solely on the biological tissue.
Right.
Yes, a neural culture processes information using local metabolic power on the scale of nanowatts.
Yeah.
It is astonishingly, almost unfathomably efficient. But you cannot run an organoid by itself on a desk.
Yeah, you can't just plug a petri dish into a wall outlet.
Far from it. This introduces what the scientific literature refers to as the life support tax.
The life support tax.
Yes. To keep that highly efficient tissue alive, sterile, and communicating with a computer network, you need commercial incubators. You need continuous microfluidic pumps constantly cycling precise mixtures of nutrient media.
And temperature control, right?
Heavy-duty HVAC systems for precise temperature and gas control. You need low-noise amplifiers, analog-to-digital converters, and massive conventional computing infrastructure just to encode the data, decode the ripples in our pond, and store the output.
Wow.
If you want to know if Wetware is more efficient than a silicon GPU, you have to measure the power consumption at the wall socket for the entire biological facility, not just the tissue sample.
So you're powering an entire miniature intensive care unit just to run a chip.
Basically.
And there are massive biological scaling issues too. Like if you want a more powerful Wetware computer, why not just grow a brain organoid the size of a basketball?
Because of vascularization.
Yeah.
Standard 3D organoids don't naturally grow blood vessels.
Oh.
If you make them larger than a few millimeters, the oxygen and nutrients can't physically diffuse into the center of the tissue mass. The cells in the middle literally suffocate, become necrotic, and die.
That is a hard biological limit.
Until we can effectively engineer artificial capillary networks, yes.
And then there is the representational drift we talked about at the beginning. This tissue is alive, it metabolizes, the synapses constantly remodel themselves.
Exactly.
This means a highly calibrated biological reservoir that perfectly recognizes Japanese vowels today might completely lose its calibration next month just by virtue of aging.
It changes.
How does a computer engineer deal with a CPU that gets tired, changes its mind, and eventually dies? Silicon is reliable because a transistor is the same today as it will be in a decade.
Exactly. Biology is inherently noisy and constantly in flux. That is why replacing silicon GPUs to run the next large language model isn't really the near-term purpose of the serious scientists in this field.
So what is the purpose? If it's not replacing ChatGPT, what is all this infrastructure actually for?
The real breakthrough is biomedical screening. Think about drug discovery. Currently, if we want to test a new candidate drug for epilepsy or a severe neurotoxin, we often put it on a static plate of cells and see if the cells physically die, or we test it on a live animal.
Right.
But with these closed-loop systems, we can use patient-derived stem cells to grow a neural network, plug it into an information processing task like balancing the pole, and then apply the experimental drug.
Oh, I see the distinction. Instead of just asking, does this chemical kill the cell, you can ask, does this chemical alter the network's cognitive ability to process information?
Exactly.
You could literally watch how a specific patient's genetics react to an Alzheimer's drug in real time based on how well their custom-grown organoid plays a game. It's a functional assay of network health, not just a toxicity screen.
Precisely. It is a revolution for precision medicine, far more grounded and immediately useful than trying to build a biological supercomputer.
I completely understand the biomedical purpose, but we cannot ignore the philosophical elephant in the room.
Here we go.
No, seriously, as these systems continue to grow, as we eventually solve the blood vessel problem, as we integrate richer sensory inputs and scale them up, we are bumping up against some massive ethical boundaries.
Yeah.
We're looking at studies showing these human neural organoids express the actual molecular building blocks of learning, markers like ARC, BDNF, and FOS genes.
Yes, the early immediate genes associated with synaptic plasticity.
The exact same chemical markers that fire in your brain when you form a childhood memory are firing in these commercially produced organoids. At what point does that fading memory in the pond become a subjective experience?
That's a lead, though.
Is it? Does it feel like something to be an organoid receiving unpredictable electrical shocks? We have folklore and spiritual traditions spanning millennia trying to define where consciousness lives. Are we accidentally engineering a new vessel for it?
I think we need to temper that.
I truly believe we need graduated oversight precautionary monitoring that scales up as the tissue's complexity increases. We shouldn't wait until we accidentally trap a conscious mind in a server farm before we start asking the ethical questions.
Look, it is a profound ethical frontier, and I hear your concern. But as scientists, we must hold the line on the evidence we currently have.
Which says what?
The International Society for Stem Cell Research, the ISSCR, is very clear on this in their current guidelines. There is zero empirical evidence that present-day systems possess sentience, human consciousness, or integrated brain function.
But they are human neurons.
An organoid is not a miniature human brain. It completely lacks the large-scale architecture, the bodily integration, the complex hormonal systems, and the lifelong developmental history of a living creature interacting with the physical world.
Okay, but the building blocks are there.
Learning, plasticity, and the presence of BDNF are necessary mechanisms for consciousness, yes, but they are nowhere near sufficient to create a subjective mind.
I hear the scientific consensus, but the fact that ethicists are seriously publishing frameworks for stopping rules debating at what point we must pause an experiment because the network is too complex shows that we are walking right up to the boundary of what we understand about life.
We are at a boundary, yes, but there's a much more immediate, grounded ethical issue that we have to confront right now, today, long before we worry about conscious organoids.
What's that?
And that is the issue of donor consent.
Ah, the consent twist. Yes, this is where the reality gets incredibly uncomfortable. Let's break this down for the listener.
Human neural organoids are grown from induced pluripotent stem cells, or iPSCs. These are often derived from the completely routine biological donation, like a simple blood draw or a skin biopsy, from a human donor who agreed to help medical research.
I want you, the listener, to think about this directly. Imagine you donated some skin cells to a university hospital for a medical study, maybe to help research a generic skin condition. Would you be okay with a tech company taking the biological descendants of those exact same cells, chemically reprogramming them into living brain tissue, and using them to build a commercial biological CPU in a server farm?
It's a huge question.
It doesn't retain your memories, obviously, but it completely retains your genome. Your unique personal DNA is the hardware. Who owns the computation generated by your living cells?
It is a massive legal and ethical gray area. The donor agreed to biomedical research, not to becoming a biological component in a commercial Wetware cloud service.
Wow.
The downstream commercial use of these biological materials has advanced far faster than our legal consent forms.
It is wild. We are literally building the future of computing out of the physical building blocks of ourselves.
If we synthesize everything we've explored today, the reality is this. We have not built conscious computers. The brain in a vat remains science fiction. But what we have successfully done is engineer living neural tissue into two-way computational loops. We have proven that biological plasticity can be harnessed to solve engineered problems in real time, opening incredible new doors for biomedical research. We have bridged the gap between silicon and synapse.
And as biology and machinery continue to merge, we are entering an era where our technology will need to be fed, medicated, and eventually laid to rest. If tomorrow's computers can grow, age, change their minds, and die, is hardware even the right word anymore? Drop your thoughts in the comments. Your perspective could help shape a future deep dive.
Share your thoughts with My Mind MashUps on our social platforms. Your perspective could help shape a future deep dive.
If you enjoyed this deep dive, like, share, follow, or subscribe wherever you listen.
Remember that My Mind MashUps is available on YouTube, Spotify, Apple Podcasts, Amazon Music, Audible, Facebook, TikTok, Pocket Casts, and other podcast platforms.
We didn't come this far just to come this far.
Yeah.
This is My Mind MashUps.